See here, it is typical eukaryotic animal cell. Here you can see membrane bounded cell organelle, that is lysosome. So lysosome were first isolated by Christian D. Dew. Dew was the first to observe such cell particles which have centrifugal properties intermediate between those of mitochondria and ribosomes. These particles, they were round in shape, dense bodies, and D. Dew, he named it as a lysosome, where lyso means digestive and some means body. So these are actually digestive bodies, contains digestive enzymes capable of lysis or called digestion. Lysosomes originate directly from endoplasmic reticulum or from Golgi membrane, and then associate with vesicles formed by pinocytosis or phagocytosis. There is evidence that lysosomes are originated from Golgi complex. Protein granules synthesized by lysosomes are seen in enlargement of endoplasmic reticulum. When these are budded up, they give rise to the bodies having properties of lysosome. Lysosomes are there in animal as well as plant cells and also in protozoa. But in bacterial cell, there is no lysosome. Under electron microscope, it is difficult to identify the lysosome because they have no characteristic shape or structure. Lysosome usually appear as dense globular body which vary in size, and size remains in between 0.2 to 0.8 micron in diameter. Lysosome is a sphere of single lipoprotein membrane containing hydrolytic enzymes which are involved in the cellular digestion. Lysosomes, they contain the substances having molecular weight higher than 200 Daltons, and such substances, they do not diffuse out through the membrane And that's why such digestive enzymes, they do not come out of the membrane of lysosome. If enzymes of lysosome, they are released in the cell, they can digest the cell, and this fear is not there because of these substances which have higher molecular weight, and because of that they do not diffuse out through the membrane and remains inside the lysosome structure and do the process of digestion or detoxification over there only. Entire process of digestion is carried out within the lysosome thus. So here in this figure, formation of lysosome from the Golgi complex you can observe. Now types of lysosome. Actually lysosomes are polymorphic, show irregularities in size and internal structure even. 4 types of lysosomes are distinguished, of which first is primary lysosome and other 3 types are grouped together as secondary lysosomes. Primary lysosome, also called as a storage granule. This storage granule or primary lysosome is small body and its enzymatic content is hydrolysis by the ribosomes and accumulated in the endoplasmic reticulum. Digestive vacuoles, also called as a heterophagosomes, are formed by phagocytosis or pinocytosis of foreign material by the cell. These are actually secondary lysosomes which contains engulfed material within the membrane, residual bodies. After digestion, the end products pass through lysosomal membrane and are incorporated into the cell. Undigested material is left, or if digestion is incomplete, the secondary lysosomes are formed and are called residual bodies. In some cells like amoeba or other protozoa, these residual bodies are eliminated. But in other cells, they remain for long time and may load the cell. Autophagic vacuoles. These are also secondary lysosomes, also called as a cytolysosomes or autophagosomes. Which are special types of lysosomes in which they contain part of the cell in the process of digestion. For example, mitochondria or portions of endoplasmic reticulum. Now lysosomal enzymes. So variety of enzymes, they are there in the lysosome. About 40 enzymes have been isolated from the lysosome. And most of the enzymes, they perform better under acidic condition, optimum pH 5. And thus they are collectively called as a acid hydrolysis. Some of the enzymes of lysosome are like nucleases. For example, acid ribonuclease or deoxyribonuclease. Some phosphatases like oligonucleotides. Phosphodiesterase. Some proteinases like cathepsin, collagenase, peptidases. Some lipases are also there, like esterases, phospholipases, glycosidases like beta-galactosidase, alpha-galactosidase, alpha-glucosidase, beta-glucuronidase, lysoenzyme, etc. Lysosomes degrade every biological macromolecule. Now functions of lysosome. So for intracellular digestion, lysosomes play a very important role as it contains many enzymes, digest material intracellularly. This intracellular digestion is of two types, autophagy and heterophagy. Autophagy means intracellular digestion in which enzymes of the cell digest the pieces of cytoplasmic material from the cell itself. That is autophagy. That means digestion of endogenous material. So by this process, mitochondria are constantly removed by the process of autophagy. Another type of intracellular digestion is heterophagy. Heterophagy means intake of exogenous material into the cell and it's break down by lysosomal enzymes. Endocytosis is another phenomenon. Endocytosis means a phenomenon in which bulk of exogenous material is taken inside the cell. That is endocytosis. Endocytosis is of two types, pinocytosis and phagocytosis. Pinocytosis means incorporation of liquid material. That is pinocytosis. And phagocytosis means intake of solid material. So lysosomes play important role in intracellular digestion. Now extracellular digestion. Lysosome enzymes are also released outside the cell and they hydrolyze the extracellular material. Thus, important for extracellular digestion. Hormone secretion. Lysosomal proteases activate the secretion of thyroid hormone in the follicles. Lysosomes play important role in the seed germination even. Lysosomes are found in plant cell. In seedlings, they are involved in the hydrolysis and in removal of protein and starch during germination. So this is as a whole the account of lysosome, one of the cell organelle.